Welcome to the OIS podcast. Today, Dr. Firas Rahal chats with Daniel Chao, Senior Director and Clinical Lead of Janssen Retina. They discuss the evolution of retina gene therapy, his work at Janssen Retina, and how his career evolved from a neuroscience researcher tucked away in the lab to playing a role in one of the leading pharmaceutical companies in the U.S. Take it away, Firas.
Welcome, everybody, back to the OIS Retina podcast. I'm Firas Rahal, a partner at Retina Vitreous Associates here in Los Angeles and a partner at Excite Ventures in New York. And I'm really happy to have my friend Daniel Chao on today. That's our guest for today. Daniel is a retina doctor himself, but he's had a lot of different hats in a fairly short time, actually. Young guy with a lot of career interesting activities up till now. We're going to hear about that. And Daniel is currently the Senior Director and Clinical Lead at Janssen Retina at Johnson & Johnson, a position he's held more recently, but he has a big background in academic medicine at UCSD, which is where I know him from. That's where I had trained many, many years ago, long before Dan arrived there. Dan, welcome to the show. Thank you for joining us for the podcast.
Thanks so much, Firas, for the invitation. Big fan of the show.
Thanks. I'm glad to hear that you've at least seen one or two episodes. That's good to know. Craig does a great job at OIS editing out all the blunders and the flubs that I create, but I'm sure that won't happen today with you. You're a very polished guy. Tell us some of your background. I shared a little bit there in a very brief way, but you did elect to go into academic medicine. Maybe you can tell us about some of your educational and training background and how that took you to, initially at least, an academic career in medicine.
Yeah, absolutely. Thanks so much for the question, Firas. So I grew up in the Washington, D.C. area and was always, I think, interested in science and medicine and putting those two together. And probably the biggest influence was my mother. She was a research associate at the National Eye Institutes of Health. And so sometimes I would tag along on the weekends when she had to go in and kind of got my first exposure. I think the next thing was then when I was in college at Virginia Commonwealth University, ended up shadowing some neurosurgeons that were there and working with them. And it was just really inspiring to see them, on one side, taking care of patients, and then the other side, then going back to the lab to try to find better treatments. And, I can certainly remember some brain tumor surgeries. They would do this very elegant surgery to really help these patients and then taking them right back to the lab to try to figure out how we can do better. And I think that really kind of inspired me to go down this physician scientist track. And moving forward, then I was fortunate to be accepted to Stanford Medical School. And while there also did a PhD in neuroscience. And, in terms of that scientific training, my PhD was in developmental neuroscience and really, I think, was inspired by some professors who were, kind of posed the question, how in nature does, the billions of neurons that you have and the thousands of connections that you make, how does that wire up and help us see, help us move, give us our personalities. And my work was not that grandiose or far reaching. I ended up studying worms, actually, round worms in particular. It's a model organism called C. elegans. And the reason why is that it really reduces that problem. Instead of a billion neurons, there's 302. And we could label the connections, the synapses with fluorescent proteins and then manipulate them genetically by mutating them or increasing the expression to understand how these proteins were, how they worked. And of course, it's not like it's completely detached from medicine, and a lot of these proteins were conserved. So then moving on, how did I get into ophthalmology? Really wasn't on the radar at all until I had a friend who was an ophthalmologist, and he was going on a mission trip to Cambodia, and he said, "Oh, why don't you come along?" And went along, and it was just incredible. I think number one, to see the amount of preventable blindness in the developing world, and seeing him do cataract surgery and as you know, literally making patients who are blind see. I mean, just how can you have more of an impact than that? And I think from there was really hooked on ophthalmology. Then went on to do my clinical training, my residency at Bascom Palmer Eye Institute, and my retina surgery fellowship at UCSF. And I think in addition to learning how to be a great clinician, surgeon, and ophthalmologist, I think I also had a lot of great mentors who were able to identify what those unmet needs were in retina, and then collaborate and build a team to actually build a product to address that and tangibly bring it to patients. And I was like, wow, that is so cool. That is, a huge impact. And how do I do that in the future? And then that ultimately led me to UCSD.
That is actually what you just last described there. That is really the epitome, the encapsulation of innovation, right? In clinical medicine, and the science of ophthalmology innovation is understand the clinical needs, understand the areas that we're weak in improving patients and their lives. And then if you have the wherewithal, go back to the lab and go back to the clinical trials and build something to address that need. That's phenomenal. And I'm sure very gratifying for a lot of people like yourself who are doing it. I'm struck by the number of people in retina who had some background in interest in neuroscience. And is that what sort of drove you to the retina space within ophthalmology? Is this kind of early interest in how neurons connect and communicate with one another? And obviously the retina is a great place to almost visualize that directly.
Yeah, no, absolutely. I think that was, I think the scientific part actually came on a little bit later. I think it was really when I was deciding, what kind of clinical specialty to pursue. Certainly was very oriented towards, the neuroscience specialties, neurology, psychiatry, and neurosurgery. And, for whatever reason, found that those didn't fit me quite well. And it was really, I think, seeing the big impact that ophthalmologists and retina specialists have on their patients, and the cool things that they're able to do. Microsurgery on one side, in the clinic, it was fun, it was interesting. The people were really balanced, happy, and really excited about what they did. But then I think, as you said, I think as I got more into the science, I was like, well, this is more or less like an offshoot of neuroscience in a lot of the ways. And as I think ophthalmology and the retina is really at the cutting edge of science with gene therapy, stem cell transplants, et cetera. So I think that certainly was an easy transition.
Yeah, we're going to get to some of those scientific ventures you just mentioned in this conversation, hopefully. And I agree, retina clinically is sort of, I hate to say this because I don't want to offend our other medical colleagues who are doing amazing stuff. It's neuroscience where people actually get better. And there's a lot of not getting better in other clinical specialties. And certainly in my time in med school, I did spend some time in neurosurgery and neurology, and I liked them a lot. But I did observe that in ophthalmology, patients got better on the whole. And you mentioned it, and I want to agree with that. That is kind of what drives
Absolutely.
a lot of clinically to it. So what did your career look like at UCSD? How were you spending most of your time? Obviously, there's this combination of clinical medicine, science, and/or lab work and teaching. And I'm sure you were doing some of all of the above. How did you view it, and what did you really like there? What were you enjoying there? What maybe didn't you like so much about a university career?
Yeah, so I think when I was finishing fellowship, I think I had the idea that I wanted to pursue a particular type of job. I think I realized I wasn't really interested going into full-time private practice or other things like that, and really focused on what's commonly known as kind of a clinician scientist pathway. And so these are more or less exclusively at academic institutions. And it's a little bit unusual in that actually the way that it's set up is that you actually spend a minority of your time in clinical care, usually about 30 to 40%. And really the main thrust of it and the expectation is that you set up a independent research program, that's funded by NIH or other foundations that's really there to advance the field. And I think that's where a lot of individuals who both really interested in science, really interested in the clinic, kind of gravitate towards. And so my time was kind of split up like that. About a third of my time was in clinic, surgery, teaching residents and fellows clinically. And then probably over half my time was then really trying to set up a research program. And for me, that was really in the basic laboratory sciences. Initially, when I started, I think along with this idea of I've kind of accumulated a variety of different kind of zoology organisms to work on. I actually started to use zebrafish to make models of retinal disease because I had so much fun playing with worms. But and it's it's interesting. I think there's a lot of parallels for anyone setting up a research lab and a startup in a lot of ways. In that I was fortunate to get some startup funding from the university, but then you have to go out there, recruit people, tell them, well, why should I work with this junior, faculty when I can go work with the Nobel Prize winner down the street? How do I get grants so I can pay these people? And how do I publish papers then to extend that? And in some ways, a very similar thought process. You have your burn rate, you're always thinking about funding and writing grants. So that was a big part. And then the other part, again, was, as you said, I think being involved in clinical trials and as well as teaching. I was the medical student director of education, so was overseeing a lot of clerkships and really the first point of contact for medical students who were interested in ophthalmology and would mentor a lot of them through that process as they were applying for residency. I think the things that I really liked was again, I think the diverse interactions of people, I had a great chair, Bob Weinreb, who was really supportive of physician scientists, lots of really interesting stuff going on, great community of clinicians. And then also just the broader UCSD community. I mean, there were so many great scientists, engineers that were doing so many cool things and got to meet some of them, work with them. We had some great collaborations. And then extending that beyond that is, the biotech community in San Diego. And so I think that was just really exciting to be able to interact with all these people. And I think the great thing about academics is that, you have the freedom to kind of do what you want and shape your career and work on what ideas you really think are important. That being said, you are kind of confined by, you need to have the financial resources to be able to do that. And I think that's one of the things perhaps I don't like is that most of your time, again, is trying to get writing grants and such. And then the other thing that can be very challenging is that, as you can see, academics, they wear a lot of hats. And how do you balance, all of those things? As being a clinician is not, you can't just compartmentalize it and say, all right, well, these are the days I'm going to do that. Emergencies come up, things bleed over. And then how do you juggle all these balls in the air?
It's a consistency across academics and the private sector, actually, that all of us who are involved in any way in development of products or innovation, raising money is always the bane of everyone's existence, getting the funds to do what you want to do. And when you get past the fundraising or grant writing part and you actually have the funds, all the fun really begins because you get to start doing the stuff you were gearing up to do. So I hear you, and that's kind of similar in the private sector. And you're in there now. Of course, you're in a different context than, say, a startup. You guys are big, and I want to come to that. But while you were there at UCSD, you already kind of started merging your career into the private sector in some ways in this whole kind of, and a lot, this is a common way things are done now in modern, at least ophthalmology innovation. Idea is sprung in a lab somewhere or a clinic somewhere in a university setting. A smart guy like yourself runs with it for a while and then maybe spins a company out into the private sector. And I know you were working with the company Visgenex. I believe you were a founder. Maybe you can share with us what that experience was like, because that seems to be, at least in your setting, a bit of a bridge and a link to have taken you to what you're doing now.
Absolutely, for us. And I would say this, as you mentioned, kind of this innovation, being involved in innovation was something I think that was sparked very early. I think probably from my medical school days, I was able to take part in this class. It's actually a business school class at Stanford called Lab to Market. Essentially, you get together in a team of engineers and business students and some engineering students, have some interesting ideas, and you work through the process of what it would be like to try and move this product forward. And so the project that I actually worked on was actually with some engineers on a glaucoma device of all things. It was a device like a glaucoma drainage implant, which had some sensors and could kind of automatically regulate the pressure that was coming out, with the ideas that, okay, we have this smart device that can control the pressure that was implanted surgically. And sounded like a great idea. The engineers thought it was a great idea. And I think I learned a lot of good things, certainly about market segmentation and not trying to develop a very complicated solution to something that is already out there. But I think going through that process just really got me excited. Well, what other ideas are out there, and how do we find something that we can move forward? And that kind of led towards, Visgenex. I think when I was thinking about how I wanted to structure my research program, it was really with that translational bent, and that I was trying to use science as a means to find some discoveries that we could then turn it into a product that can help patients, versus being perhaps just more, I'm interested in it for the sake of discovery and science. And so it turned out for this particular project, my initial research was kind of stalling, and I was thinking like, well, how do I kind of pivot into my next chapter? Part of it for my academic survival, because you always need to bring in grants and such, and also I think
perish, isn't that?
Yeah, it publish or perish, and I think part of it was also, what kind of impact would I have? And I remember one day kind of sitting in the lab, and we're doing drug screens and zebrafish, and I thought, oh, we're going to discover new drugs that could help people, but I think what I came to realize is that was just so far away from actually reaching patients. And I'm sitting there going back, all right, I got to go to the microscope room, look at all these zebrafish, and like, is this really the best use of my time? And is this something I really want to keep doing year after year, in terms of making an impact? And I think obviously the answer to that was no. But going back to Visgenex is that I think this was another chance happening, is that one of the other junior faculty in UCSD, a PhD basic scientist named Dorota Skrounza-Krauschek, who's now at UCI. She just made a really remarkable discovery, linking this lipid enzyme that had actually been where the gene regulation of the methylation of this lipid enzyme was actually a great biomarker for aging. And some people actually use it for like forensics to determine age of individuals. But she discovered basically that it seemed like when you eliminated this lipid enzyme, it would actually accelerate aging in the retina and also give some phenotypes that resembled AMD in a mouse. And hearing about her work, and I was like, wow, just it was clear what the possibilities were from a translational side. And I was like, I'd love to work with you together to move this forward and take advantage of her scientific expertise, my clinical expertise, and we started this great collaboration. Scientifically, we were able to get a number of grants, publications, but I think also able to spin out this company, Visgenex. And it was kind of a funny story. After we had started collaborating, we were thinking, all right, well, let's work on the science a little bit to have something more solid before we really think about spinning it out of the university. But it turned out there were these kind of biotech executives. They were kind of looking for their next thing and went to the tech transfer office at UCSD. And, they said, oh, do you guys have anything interesting there? And he said, oh, why don't you take a look at this stuff in the eye? And they really liked it and were really interesting in spinning it out. And we kind of said, yeah, let's get together, let's partner up and out the company. And that's what had happened. And as we move forward, then was able to raise some seed money and got us off and running.
That's fascinating, that part of it. I didn't know that part of it. I'm fairly familiar with the tech transfer and spin out concept, and often it's in the other kind of direction, which is the innovator is then sort of pursuing this, what you're describing. And I know of this too, because people who are in incubators do this, and then maybe some execs who are looking for the next best thing. You're sort of describing a scenario where these folks were just sort of looking and asking the tech transfer people to tip them off to something that might be of great interest. Is that common in your experience, or is it usually the other way around?
I think this was pretty uncommon. Usually, it is the other way around, where the academic inventor really invested. Usually, that applies. There are kind of these NIH grants that help kind of start up and translate things,
Yeah.
kind of move it along to the point, and really are the champion until they can recruit someone like a professional then to help manage it and really move it forward. And I think there's pluses and minuses to both ways. I do think, though, that the earlier you partner with some experienced business drug developers, is that it certainly helps to greatly accelerate the whole process in terms of raising money, in terms of really figuring out, all right, well, what are the next things that we really need to do? Also understanding manufacturing, other things that really you don't really think about as an academic. But it can be very tricky in terms of the partnership and making sure it's the right collection of personalities. Obviously, the academic inventors are like, this is the best thing since sliced bread. Why aren't people giving me money? Why don't you see that this is great? But then also, like, I think the business people, they can sometimes then, as they get further along, then start to exclude the scientific founder, and as they move things forward. And I think fortunate ours, we were in a team that kind of clearly recognized everyone's area of expertise. They didn't really understand ophthalmology or really the deep science behind it. And we were naive kind of to the whole drug development process and how to raise money and move things forward. So I think it really ended up being a good partnership in that way.
Yeah, it really sounds like it. And I think you were fortunate with the type of people you connected with. There's so much there that we wouldn't have time to review, unfortunately, because I love this topic. And for people who are listening, and a lot of our listeners might be young entrepreneurial type academics who are exploring these options, and it's a little intimidating, and even striking that first deal in the case that you just described. If someone like yourself who hadn't done it before, you can end up in kind of maybe not the best situation, but it sounds like yours worked out very well. And you had the right partners, which is the key. I agree 100%, is who are these people that you're partnering with? What are their short and long-term motivations and their ability to raise money? That's really critical part of it. Most scientists aren't really geared towards fundraising. They don't want to spend their time doing that. They may not be particularly good at it. Did the tech transfer people help you at that time and guidance, or was it really just you and or any private advisors that you got?
They were actually very helpful. I think the UCSD Tech Transfer Office is very innovative friendly. They really had to push to get their things out there. And I think our particular tech transfer officer was great. He really worked with us hand in hand in terms of thinking about filing the provisional patent, and was really a great champion of our idea and product. We were able to get some internal funding from UCSD to push it along. And obviously he was a big advocate to also external partners as well. And so I think really when you're working with tech transfer offices, I think each university kind of has their own attitude and philosophy, and some can be very difficult to work with, and some can be very more friendly. And I think it's talking to other individuals at your institution who've really navigated all of these things, and who should there's probably better trans tech representatives and not, and really having someone who's done it at your institution that can really help you to navigate that.
That's good advice. I don't want to get into the details on Visgenex because I think it's fascinating science, but just to use it as a segue into some of the broader scientific innovations we're looking at now in retina, Visgenex has sort of becoming, if not initially, a gene therapy company, as you just described. And there's so many different angles on gene therapy. It's not just what people think of overly simplistic is replace a gene with a gene. There's a whole lot going on there. In that context, what do you think about retina gene therapy on the whole? And I know this is probably a 10-chapter discussion, but is this, our field was revolutionized with anti-VEGF therapy 16 to 18 years ago. And we know this, and everyone in our business knows this, especially those of us who are around before that. Is this now that level of explosion? Is that what's going to happen here? I mean, how do you view retina gene therapy? Or is it just going to be an incremental advance? Or is this revolutionizing what we do?
I think we're in the early days, and I think, like you, very excited about gene therapy as a platform, and the potential what it can offer. As we know, we've really seen some breakthroughs with Luxturna really demonstrating that we can deliver gene therapy, that it's more or less well safe and well tolerated for the most part, and actually bring clinically meaningful improvements in vision in some of these genetic diseases. And then, in other approaches that are using gene therapy as a biofactory, that it can produce therapeutic proteins for long periods of time that can have significant biological effects. So I think it's really exciting, and the idea of having this one and done therapy is very attractive, and kind of the ultimate goal. In my personal opinion, not really that of Janssen, is that super exciting, but I think there's also a number of challenges that the field overall needs to overcome. And I think there are three things. One is the inflammation, or what we call gene therapy induced uveitis, that we've seen in a number of programs. Clearly there's an immunogenic response in the eye, even in this immune privileged area, and that effect can be quite varied. But we really don't understand it from a scientific level. What are really the factors that are involved? How do we design better vectors or capsids that help to minimize this inflammatory reaction? And then, how do I identify patients that might be more susceptible to this? So I think that's something certainly that as a field we'll know a lot more about. I think the second thing is also the manufacturing of gene therapy, and that it's quite new. Currently, it's it requires a lot of work, a lot of money, and really the process from the different cells that you use, the different plasmids and vectors. I think all of that is still being optimized. And then also, how do you scale that to larger quantities that you would need? So I think also that's moving very, very quickly in the field. And then thirdly, I think pricing. How do we? I think we're just seeing with Luxturna, how are we going to price this? Right now it's thought, these are truly one and done therapies. And the way that insurance companies are looking at it is really like kind of a pay for performance. Like, well, if it's working this year, all right, we'll pay for it. But I think we'll really have to see long term, can gene therapy really deliver on its promise? And I think we're in this kind of generation 1.0 for gene therapy, but certainly very excited as the field continues to move forward, is that will really make it, has the potential to be this great platform, certainly for a variety of retinal diseases.
Yeah, Dan, I totally agree with all of that. And we are here at Retina Vitreous involved in a lot of these clinical trials. And there have been, as you alluded to, some cautionary tales regarding inflammation, something that's always been a concern actually right from the beginning, but it was theoretical then. And now that we have actual clinical trials and early clinical data, that is going to be one of the largest challenges, obviously separate from efficacy, but the inflammation is real and there have been some problems, but I'm sure smart guys like you will work that out, which brings me to the sort of final topic. And you kind of alluded to it. You're now at Johnson & Johnson. You're in the private sector. You work in the industry as an executive. It's a great position. And you might view things differently. You might not. Tell us about the position you're in now. What attracted you to it, and what are you actually doing on a day-to-day basis in the short term anyway?
So maybe I'll start with that part first. So my position, I'm a clinical lead within our translational and experimental medicine group. And so that's really focused on early stage clinical drug development. So my primary responsibility is really to design and execute those first in human phase 2 proof of concept studies for assets that are moving out of our discovery group into the clinic, or potentially also working with external partners. And along with that, that's also involved in, for instance, developing biomarkers, which may help us understand how these drugs may be working, and how can we accelerate our ability to make decisions on these clinical trials, make trials shorter, use for your patients. And for that, doing a lot of work, for instance, in data sciences, big data, imaging, and it's been fun because I've been able to work with a lot of my previous academic collaborators in that realm. And then I think the third part is, I think, related to that is then also talking with external partners, hearing about their technology and, just helping to support decisions and potential partnerships that Janssen may be interested in. So I think it's a really incredible, that was kind of all the things I was really interested in terms of going into industry. And so, really fortunate to have this position and really, really enjoying it.
That's a big question, but
yeah,
honestly, with regard to J&J and in your current position, is the Retina division new? Is this something J&J has been doing a long time? Or is this a relatively new space for J&J in the Retina space?
It is relatively new. And I think that was one of the big attractions for me. So about 3 years ago, J&J made the strategic decision to get into the retina area and develop a therapeutic area from scratch. And when J&J decides to do something like that, they really invest the resources and have that commitment and ambition to really be a market leader in that area. And so that's been very exciting as I think the team is still relatively small compared to, let's say, other pure pharma companies, but really moving very quickly and growing. And I think just in the past 3 years have developed a number of clinical stage molecules. And I would say really the goal of Janssen is really to identify, what are the not incremental assets, but really transformational assets that are really going to bring significant value to patients. And that's across both rare disease as well as common diseases. And certainly at Johnson & Johnson, fortunately, we're able to have access to all the different modalities, gene therapy, small molecules, et cetera. So we can really figure out find the right tool for the indication. And I think the third thing is that I think Janssen and Johnson & Johnson is a very innovation agnostic attitude. In terms of we understand that we're doing a lot of great work internally, but can't do everything, and that there's a lot of great external partners who are doing really great science, and we're trying to be part of that ecosystem, and if we can partner up with groups to help advance those products and really turn them into things that can help patients.
So I think you clarified this, but I'll just for clarity for the audience members who have great interest in that point, your company then is interested in both internal development and potentially also acquisition or partnership as a direct route to these treatments that you were just describing.
That's right. I think if you look at Janssen across the board, is that again interested in both of those avenues, and I think also at what stage you interact with a company like Janssen, and I would say at any stage, I think we're extremely flexible in terms of the type of collaboration, whether that's kind of an academic research agreement versus co-development versus licensing versus acquisition. I think really focus on finding, what's the best thing to make it a win-win situation for all parties.
That's great. I'm glad you covered that. That's often of great interest to some of the startups who are listening and innovators who are listening and want to know how to proceed. In our final couple of minutes, back to the personal part, which you were about to allude to, about the decision to make the leap, and one, how'd you make it? Was there apprehension? I'm sure there was. We're all professionals. There's always some apprehension about going into the unknown, but specifically, how did you view that then and now? And do you already, or do you foresee missing clinical medicine? I'm sure you've thought about that as well. Taking care of patients has a certain unique kind of quality to it that there's not much else like that exactly. And finally, how would you advise others in your previous life who are considering such a move?
Yeah, for us, and I think we could talk hours about that, but I'll spare you that pay to give you the short points. But I think when I was about 2.5 years ago, I really wasn't thinking about this at all, wasn't on the radar. I think my academic pursuits were finally really getting off the ground. But I think at that point started to hear about different opportunities in industry and pharma, and was certainly very curious and check things out to really understand what's involved in drug development. And I think when I ended up talking to people, thought, hey, this is a pretty cool job. I mean, really smart people working to try to, again, really motivated by finding things that can help patients, and work was really interesting. A lot of similar peers. There's a lot of physician scientists who end up going into and pharma. I was like, okay, this is really interesting, but not for me. Maybe I'm pretty happy with what I'm doing right now. And it's interesting. I don't know if it was midlife crisis or kind of existential deliberation of what I'm doing. But just thinking long term, more 5, 10, 15 years down the road, like what do I really want to be doing? And it was very clear from an academic standpoint what that would look like. And it would be, a lot of it would be involved writing papers and writing grants, and realized I'm not sure if that's really what I want to do for the rest of my life. And when I really kind of thought about, all right, well, what do I really enjoy the most about my job? And it turned out that was actually thinking about a really interesting discovery or technology, getting to a group of a small group of people together and figure out, how do we move this forward, either into the clinic or beyond. And I realized that was really what, again, kind of drove me the most and what I was most excited about, and really realized that, hey, that's what they do in industry as a kind of full-time job. And so that got me thinking, oh, maybe I should think about this more. Had a lot of conversations with mentors, that kind of asked, well, am I crazy for jumping and doing this, giving up all the training that I've done, and have a pretty good gig right now, because as you know, few things are better than being a practicing retina surgeon. But it's interesting, even those that were, I think, really hard fire in the belly academics, they were actually all kind of very supportive. I think they had probably seen what kind of the potential of what one can do in industry, and it was kind of quite interesting. They were very supportive, and I think this was about a year-long process, and I think at that point kind of realized, okay, well, if the right thing came along, I would seriously consider it. And certainly when this Janssen opportunity came around, I was like, oh, this is an incredible opportunity, and decided to take the leap. And it's been about a year, so still pretty early into it. Do I miss clinical medicine? Yeah, there's certainly a certain part of me that does. Being able to treat patients, do surgery. But I think I also really enjoy what I'm doing now, to be able to have that larger impact and think about these larger questions. And so I would say I'm happy with the decision that I've made. I don't regret it, and really looking forward to what happens next.
I think it was really rational, and I want to repeat it because I really like this part of your answer, that we should all be thinking about what part of my day, what part of my job do I really like the most. That's kind of it in a nutshell. And if we all do that, we might make various decisions based on that. I think, given what you just said about what you were enjoying the most at the university clinical scientist practice that you ran, very successfully, I might add. I know about your work before. And that was the part that you liked the most. So why not? I congratulate you for it. I think it's really an honest answer and the right answer for anyone else considering it. They ought to do that inventory that you just described. And before I conclude, I want to say one thing. If you're in midlife, I need to check in for Social Security soon. I know, for all those listening who are more in my age group, that's Dan's version of midlife. Thank you, Dan, for joining us. That was really, really great and educational for me, and enlightening about how one goes through the different options in science and clinical medicine and university practice, et cetera. And I congratulate you on your leap to your new career. I'm sure you're going to do amazing things, and you and I will have an opportunity to work together as we have in the past.
Yeah, Faraz, thanks so much for the invitation. Always great to speak with you, and looking forward to future interactions in the future.
Thank you. Thanks very much.
Thank you for listening, everyone. We hope you've enjoyed this episode of the OIS Podcast. Be sure to listen in next week as we discuss the latest innovations in ophthalmology with experts in science, medicine, and industry. Subscribe to our iTunes channel so you don't miss a thing. Got a story of your own to tell? Apply to be a guest at ois.net.